Hello everyone, I hope you all are doing great. In this video, we will be studying about protein synthesis. There are three steps in protein synthesis. We will be studying in detail about these three steps. Steps are transcription, translation, and translocation. Before starting about protein synthesis, let us first understand the difference between DNA and RNA. We have studied about DNA and RNA in 9th standard. This is just a revision of DNA and RNA. So, DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. DNA is double-stranded. RNA, on the other hand, is single-stranded. There are 4 nitrogenous bases in DNA as well as 4 nitrogenous bases in RNA. Now, what is the main difference between these 4 nitrogenous bases? In DNA, we have adenine, guanine, cytosine, and thymine. But in RNA, we have adenine, guanine, cytosine, and uracil. Here is the main difference that in DNA, we have thymine, which is replaced by uracil in RNA. Now, the DNA is found in the nucleus of our cell, whereas RNA is found in the nucleus as well as cytoplasm. Also, certain portion of the DNA codes for the gene, and when a gene is expressed, a certain type of protein is produced. Now here, I have added a table for you to differentiate between DNA and RNA. You can pause the video and note it down. Now, there are three types of RNA. First is mRNA or the messenger RNA. Now, what does mRNA do? It is like a postman. It carries message. Now, what it does is it carries the information of protein synthesis from DNA in the nucleus. This is very important. From where to where it is carrying the message. From DNA in the nucleus to ribosome in the cytoplasm. Now, if you don't know what are ribosomes, so ribosomes are organelles in the cell. It is present in the cytoplasm where the synthesis of protein takes place. The other type of RNA is tRNA or transfer RNA. It is like a hairpin. Now, what the tRNA do is, according to the message of mRNA, whatever the message mRNA has brought, the tRNA carries amino acid up to the ribosome. So what it is doing, it is carrying the amino acid from one place to the other place. Next type is rRNA. It is the component of ribosome, and ribosome performs the function of protein. Now students, you may think, why are we learning protein synthesis? Why they are so important for you to study? So well, proteins do all kinds of work in our body. Example, in transport, in making structure, in acting as enzyme, in protecting our body, as well as proteins are essential for us to live. In fact, your body is making proteins right now as you are watching this video. So let's start with protein synthesis. Well, you may also think how proteins can be made from DNA, which is in the nucleus, as protein can be made by ribosomes, which is in the cytoplasm. How does this transfer takes place? Well, here comes RNA to the rescue. It plays a very important role in this process. So, proteins are synthesized by DNA through RNA. This is called as central dogma. Here in this picture, you can see that DNA has specific nucleotides of genes, and then through DNA, mRNA is transcribed, and from the mRNA, proteins are formed. Further, we will understand how proteins actually are formed with the help of three steps. So, the first step in protein synthesis is transcription. Now, transcription is a process in which we are going to transcribe the DNA into a message. Now, how are we going to transcribe into a message? So, in this step, an enzyme called RNA polymerase, it comes and binds to a specific sequence. As soon as it will bind to that sequence, the two strands of DNA breaks. One strand will be the template strand, which will be used to generate the mRNA, and the other will be the non-template strand or the coding strand. So now the RNA polymerase move along the template strand and it starts to read and to produce mRNA. The only difference between the mRNA and DNA is of uracil. That is, uracil will be present in mRNA and DNA will have thymine. You can see in the picture that mRNA is complementary to the DNA strand. The mRNA which is formed now carries with it the code which will be in the DNA, and the codes are for amino acid which consist of three nucleotides. Now, as in the picture you can see there are three nucleotides assembled together. These are called as triplet codon. Now, as the mRNA is formed, the RNA polymerase detaches itself and the DNA zips back as it was. Now the transcription is completed. The DNA has returned to its original state and our mRNA is produced. Now the mRNA has the message for the proteins which it has coded from the DNA. Now the mRNA leaves the nucleus. You will see in the picture the mRNA is leaving the nucleus and it will move into the cytoplasm where it will find a ribosome. This is where the translation occurs. Now the next step is translation. During translation, the mRNA act as codes for specific protein. And now, inside a ribosome, the ribosomal unit binds to the mRNA. Here, the tRNA, according to the message on mRNA, will bring the amino acid. So, the amino acid are brought by tRNA. This is called as translation. If you will see in the picture, the second picture, the tRNA is bringing different amino acid according to the message on mRNA and is dropping that particular amino acid and leaving. So the tRNA drops the amino acid and leaves the ribosome. The process continues till all the codes of mRNA is read. Now, during the process, the ribosome keeps on moving from one end of the mRNA to the other end by the distance of one triplet codon. This is called as translocation. So many chains of amino acids come together to form a complex protein and are attached to each other by a peptide bond. This is how proteins are synthesized. Now, the characters are transferred from parents to their offspring via genes. But sometimes sudden changes may occur in those genes. Even a minor change in any nucleotide of the gene is nothing but mutation. Mutation may cause genetic disorders like Down syndrome, sickle cell anemia, etc. So students, in this video, we studied about DNA and RNA, the difference between DNA and RNA, the different types of RNA, and about protein synthesis, which includes three steps: transcription, translation, and translocation. Now here, I have mentioned few questions. Try to answer them. And if you can't, try to find the answer from your textbook. What is central dogma? Define transcription. Define translation. Define translocation. What is triplet codon? What do you understand by the term mutation? Explain the process of protein synthesis. So, I hope you all understood the topic. Thank you for listening. Until then, take care of yourself and stay safe. Bye bye.